Would like to thank the organizers for inviting me in this exciting meeting. And I'm going to speak about next generation sequencing for diagnosing infectious diseases. But before I do that, I would like to disclose that I have been employed by ID by DNA and that I'm currently expert advisor for ID by DNA, a company that is designing software to analyze metagenomic data. So, application of next generation sequencing in clinical microbiology and infection prevention has already been introduced several years ago, and it's getting more and more popular. Whereas it started with looking at infection prevention and measures and outbreak detection, it's more and more used for really diagnosing infectious diseases. And actually, maybe we can see next generation sequencing as a one-stop shop in clinical microbiology. The different steps of next generation sequencing are quite similar to the steps that we perform in the traditional microlab. So we start with preparing the samples, and then we can use next generation sequencing for identification of all pathogens, for antibiotic susceptibility testing or predict the resistance against antimicrobials. We can also use it for the detection of virulence and toxin genes, and we can use it for typing. I will not speak about typing because I do that in another presentation later during the meeting. So why do we need such modern technologies? And are we actually really needing them? Well, if you look at the current diagnostic capability, then it's obvious that we still fall short here. And in many cases, we do not come to a diagnosis, or we fail to obtain the etiologic diagnosis. In 80% of encephalitis meningitis cases, 60% of pneumonia, or 40% of sepsis, we do not know the etiology of the infection. And this results in an extended intensive care unit stay, more anti-infective usage, and also increased costs. So we need something new there. And can clinical shotgun metagenomics, for example, be a tool to use to improve our diagnostics? Clinical shotgun metagenomics, obviously, when you want to apply it to clinical microbiology, you should make sure that you take all the kind of controls into account that you also use for other molecular testing, like a positive control, negative control, and so on. And also the use of an internal control is very important and allow us to do quantification, as I will show later. So clinical shotgun metagenomics is sequencing everything that is present in a sample of a patient or animal or the environment. It starts with extracting RNA, DNA, making your cDNA DNA library. So you have to convert your RNA first to cDNA when you want to detect RNA viruses, for example, and then sequence simply everything that is present. This technology is able to detect any pathogens, and it generates a lot of data. Therefore, we need to prioritize the results before we put them in a laboratory report and hand them over to a clinician. Is clinical shotgun metagenomics already really possible in clinical microbiology? I think it's really a discovery and a breakthrough in discovery and detection using this sequencing technology and analyzing microbial DNA. And actually, when you compare the time it takes, then conventional testing takes already longer than shotgun metagenomics. I mean, we can generate data already within 24 hours, and this includes the analysis of the data. Analysis is actually not an issue any longer. It takes only 5 to 10 seconds to get the whole results available. So 24 hours for clinical metagenomics compared to conventional microbiology, that can take to a few days. And when you first have to culture your bacteria and then do the identification, do the antibiotic susceptibility testing, and so on. This can all be done with metagenomics, or at least we can predict antimicrobial resistance. And to show you that this is really helpful, I have a use case here where we had a Prevotella pleuritis infection diagnosed in a 12-year-old person. This 12-year-old male had a history of obesity, type 3 diabetes, and he presented to his primary care provider with right-sided pleurisy. So the chest X-ray showed a lower lobe infiltration, and the boy was admitted overnight and given some antibiotics. Diagnostics was performed. A respiratory PCR using a viral panel indicated the presence of human coronavirus OC43. However, this was not really thought to explain his lobular pneumoniae. But since the boy mildly improved, he was discharged the following day. Unfortunately, 3 days later, the patient returned to the emergency room with worsening symptoms, and he was again admitted. So again, pleural fluid was taken, and an aerobic and anaerobic culture was done. And these results turned out to be negative. Then the sample was also sent to our lab to perform clinical metagenomics. And we found a Prevotella pneumoniae, a rare pathogen that only recently has been recognized to cause these type of symptoms. So immediately the patient was placed on anaerobic coverage with some other antibiotics, meropenem, and then improved markedly. So this shows that clinical metagenomics is an unbiased way of looking at the presence of pathogens and is able to detect co-infections, as we also detected human coronavirus OC43. Now, this technology can not only be used for respiratory infections, we can also use it for diagnosing urinary tract infection. And in this case, we tested 115 culture positive and negative routine urine samples and performed again shotgun DNA sequencing. As you can see in this graph, we were able to identify and bacteria in 95% of the samples that were also positive with culture when the load of the bacteria was over 10.5 C colony forming units per mil. And this is kind of a limit that is used in clinical microbiology, because when you have a urine and you have less than this 10.5 colony forming units per mil, we sometimes say that the bacteria is just present and it's not causing an infection. So good result here, but we also see that 3 samples with urine culture with loads over 10.5 CFU per mil were negative with shotgun metagenomics. Now let us look into more detail into those samples. So when we troubleshooted these 3 urines that were positive in culture but negative in our sequencing approach, we found actually that in all 3 samples, the microorganisms detected by culture were also present by next generation sequencing, but they were not called positive because they were below a certain threshold that we always apply. And if we look into more detail, then we see that in some cases, E. coli was not reported because it was dominated by human DNA. It was more or less outcompeted by the host DNA. And in another sample, we found that Klebsiella pneumoniae was present, but was dominated by anaerobic bacterial species. So it was outcompeted by non-clinically relevant flora that was present in the sample. So this is causing a problem, and it causes that our sequencing approach may be not sensitive enough. However, for that, there are options to improve that. I show you here a slide showing the spectrum of molecular diagnostics, including PCR and multiplex PCR. But let us focus on the sequencing-based approaches. There are mainly 3 flavors, that is amplicon sequencing-based amplicon-based sequencing, shotgun metagenomics, and then precision metagenomics, in which we make use of hybrid capture probes. Now, whereas amplicon sequencing allows you to be sensitive, it has only a limited breadth. Moreover, it has a low mismatch tolerance. Shotgun metagenomics is very deep, but it has variable sensitivity, and especially for the detection of antimicrobial resistant genes. Now, precision metagenomics is very broad, can detect all types of pathogens, and can be sequenced at moderate depth because it is very sensitive and specific, and also reliably detects antimicrobial resistant genes. Let me give you an example of precision metagenomics. Precision metagenomics starts similar to clinical shotgun metagenomics. So we extract RNA DNA, make use of the positive negative control and the internal control, and then we prepare the libraries again. But now, before sequencing, we enrich the libraries with hybrid capture probes, targeting the pathogens of interest. And then after enrichment, we will sequence the libraries. And then we detect enriched pathogens. Again, we need to prioritize the results and generate a laboratory report to be able to inform the clinicians. Let me give you an example of a panel that is brought on the market by Illumina, co-designed by ID by DNA. And this is a respiratory panel that identifies 187 bacteria, 42 viruses, and 53 fungi. And in addition, it also includes antimicrobial resistant genes, including the antiviral genes. So you see here in this slide, the top targets on the respiratory pathogen ID AMR panel, and it includes, for example, Bordetella pertussis, but also Mycobacterium tuberculosis. As I told you, we have many genes are detected that cause antimicrobial resistant genes, and this includes not only antibacterial mechanisms, but also antiviral and antifungal ones. Now let me show you an example of the performance of this panel. In the next slide, you see in the left column, here we used patient samples that have been analyzed previously with multiplex PCR. And here you see that when we use shotgun metagenomics, we still miss several of the samples, pathogens that were detected by real-time PCR. However, after enrichment, we saw that the detection of various respiratory pathogens in these clinical sample libraries was significantly improved. And we identified all the pathogens also detected by real-time PCR. When we look at the fold enrichment, and that is shown here in this slide, so on the x-axis, you see the pre-enriched libraries, the number of reads we find, and on the y-axis, you see the fold increasement after enrichment. And enrichment was most pronounced in samples with lower pathogen loads. When we look a bit further, we can do more with these kind of technologies, because as I told you, when we add internal control into our clinical samples, we can use this to also absolutely quantify the detected microorganisms. In general, pathogen detection is based on reads per kilobase million, RPKM, a metric that normalizes the number of detected reads to 1 million and then to the length of the targeted region. Now, by using RPKM and this formula indicated here in combination with spiking a known amount of an internal control, in this case T7 bacteriophage, we can simply calculate the absolute quantification of the pathogen detected. And that this really works is shown here, where we did this for SARS coronavirus 2. Here we compared SARS-CoV-2 quantification by qPCR with precision metagenomics. So on the x-axis, you see the viral load as detected by precision metagenomics, and on the y-axis, the viral load as detected with real-time PCR. And there is a huge correlation between the viral load detected by the both methods. There is, however, an advantage of using precision metagenomics for quantification, because now we don't need to generate a standard curve all the time. So we can just simply use the formula I just showed you to determine the pathogen load. Now, since SARS-CoV-2, the whole genome is covered by this enrichment panel, we are also able to type the virus, and that is shown here. When we looked at the different samples indicated on the x-axis, we were able to show the pango lineage to which the virus belonged to. So quantification, detection, and typing is possible by using precision metagenomics. Now, obviously, with this increasing complexity in diagnostics, and this already started with, let's say, the panel PCRs, with more than 40 pathogens now and then, we should not longer think like, use the pathogen-specific thinking, but we need to think in syndrome. What is the impact for the patient? Obviously, there is a challenge on how to transfer the information to the physician, and what is the impact? And obviously, it comes with higher cost. And this is especially also true for metagenomic sequencing, either shotgun metagenomics or precision metagenomics. So how do we kind of report the results? Well, it starts all with having a diagnostic grade database. So there is a lot of things that we detect, a lot of microorganisms, but we need to fish those out that are clinically relevant, and then maybe even go to the ones that are known to cause diseases. Now we can combine already existing knowledge in literature and apply this by using all kind of artificial intelligence, machine learning methods to come to kind of train the software to report the detected pathogens in a way that it makes sense for the clinician. So, as I told you, as we detect so many pathogens, it's really necessary to distinguish, let's say, the microorganisms that are just present from the ones that are really the pathogens. So making use of thresholds that will determine that a relevant pathogen was detected is an option to do that. So the optimal threshold will depend on the application and required confidence. So we can work with thresholds, and that is also what you can do in your software. You can classify the detected pathogens based on the general classification with normal flora, and those that are known to be associated with disease. And then, for example, come to a score in which you say, okay, this is phenotypic group 3. This is really normally not part of normal flora, but really causing the disease. So reporting and interpreting phenotypic groups may be helpful to transfer the results obtained by these modern technologies to your clinician. Another interesting thing is that, and I have to stress before I continue, that obviously this phenotypic group does not indicate pathogenicity in any case. And of course, the results need to be interpreted in the context of all the other available information. Another interesting approach is because with metagenomics, as I didn't tell you, but we do not only sequence pathogen DNA, but also the host DNA. Now, can we use the sequencing of the host DNA, RNA, to make a prediction of what is causing the disease? So can we integrate host response and unbiased microbe detection for lower respiratory tract infection diagnosis in critically ill adults? This is a paper in Proceedings of the National Academy of Sciences by other authors. And here they wanted to distinguish the pathogens from, let's say, commensal or colonizing microbiota from the ones that are causing lower respiratory tract infections in respiratory samples using metagenomic data. And what they did, they used RNA sequencing and DNA sequencing of the samples. This allows, as I told you, simultaneously host transcriptional profiling, but also detection of RNA pathogens like RNA viruses. And also, interestingly, it enriches for actively transcribing microbes versus the ones that are latent or non-viable anymore, because in that case, you will only detect the DNA. Now, by doing this and by using two kinds of machine learning models, they try to distinguish probable pathogens from airway commensals. And they use two different models indicated over here, a rule-based model and a more flexible logistic regression model. That also, the latter also permitted detecting novel pathogens, whereas the first one detects more the well-established respiratory pathogens. They looked at three outcome metrics. One was the pathogen detection, the second was the lung microbiome diversity, and the third one is looking at host transcription factors, indicating in this picture on the right, you see a list of the transcription factors the authors have been looking at. And they found that both the rules-based model and the logistic regression model achieved accuracies over 95% to differentiate pathogens from respiratory commensals. When they looked at the different outcome metrics, they found that the pathogen metric had an area under the receiver operating curve of 95%. So this performed more or less the best. The lung microbiome diversity metric had showed the least, I would say, with an area under the curve of 0.8. So the host transcriptional classifier had an intermediate performance. However, interestingly, when they combined the outcome of these three metrics, they achieved a negative predictive value of 100%. And also predicting in an accurate way with 100% accuracy, a negative result is extremely important for treatment or non-treatment. So very promising results here. So in my opinion, when we look at the future of clinical microbiology, I'm quite convinced that modern technologies will start to replace existing diagnostic methods. And since we generate so many data, it's simply not possible any longer to do that by heart. So we need more and more computer-based algorithms to interpret the results we obtain with these new modern technologies. I'm convinced that by doing this, we will go from simply detecting a microbe to really true diagnostics, which will be very beneficial for patients. Now, I want to challenge you. In the future, will there be a reduced role for the clinical microbiologist? Secondly, will there be an increased role for a molecular microbiologist? Or it's clear that maybe in the future, clinical microbiology will be heavily depending on bioinformaticians. And I invite you to discuss this with me. I'm very curious about your opinion on these statements I made here. And with that, I would like to finish my presentation and show you the collaborations I had over the years with many people in many different countries. And I would like to thank you for your attention, and I'm happy to discuss the statements I just made with you. Thank you very much.
Hello. Good morning, Professor Rossen. My name's Barnaby.
Good morning.
Yunn. How are you? It was a very interesting talk, thank you. I wonder if, before discussing the role of a clinical microbiologist, molecular microbiologist, or a bioinformatician, I could just ask you, we had a talk earlier on from Professor Niranjan who was talking about quasi-metagenomics for environmental microbiome samples. So collecting samples and doing some initial culturing and then sequencing it. Is that something that would make sense with clinical samples so that you sort of can fit somewhere in between unbiased metagenomics where you just sequence everything versus precision metagenomics? You do some preliminary culture to get good quality whole genomic samples.
Yeah, thanks for this interesting question. Obviously, that is a possibility. However, that will introduce a bias again, and that is actually what we want to try to get out of the diagnostic process. So, if you do a culture step, then you biased to those samples that are viable and that are easily culturable. So, for example, TB takes a long time to culture. Versus Staph aureus that has a much faster replication cycle. So you will more replicate Staph aureus versus TB, and therefore you get a bias of your results. Secondly, of course, you will not find your viruses, as those cannot that easily be cultured. The other thing is that in clinical microbiology, sometimes a patient may have already been treated with antibiotics, causing that such microbes are not replicating or not growing any longer. Again, you may not detect them because those will not be, well, let's say, enriched by this culture step. So you can have a try, but I'm afraid that in clinical microbiology, you will bias towards those microorganisms that are easily replicating versus the ones that are not replicating at all, including viruses.
Yeah, that makes sense. Okay. So we have a question here, which from an anonymous person, perhaps defending the clinical microbiologist, I don't know, asking what, in your opinion, are the barriers to adopting this clinically? Is it access to technology? Is it the costs of metagenomics or sequencing, or how to integrate it into current workflows? What's going to stop this happening, yes. I guess?
What's going to stop this happening? For now, I think the biggest challenges are still mainly the turnaround times. Although we are having them in 24 hours, I mean, compared to bacterial culture, this can be faster, as I told you. On the other hand, if you, I'm working in a hospital now, and we see COVID cases rising again, and on the emergency wards, they just want to have this point of care testing devices that can give you a results already in 10 minutes. Well, that is currently not possible with metagenomics. So we definitely have to work on the turnaround times, and with technologies as, for example, Oxford Nanopore technologies, this may be already occurring. So you can already see the results during sequencing, but still you don't have the results within 10 minutes. So we have to work there. I do agree that in most cases, sequences are not randomly accessible. So that means that you have to sequence batch-wise. So that is something we have to work on. So I think those two, in combination with obviously the complicated results that you get, are some barriers before, that we need to address. Still, I think that we can already introduce it in clinical microbiology and slowly, hopefully work on the challenges that it still brings.
Okay, thank you. And a question here from Gabrielle Yunn. One of the issues with the use of unbiased sequencing, as you did touch on, was determining what is signal, what is noise. And is that something that needs to be worked out for each individual sample or each individual illness? And what is the best determinant to use? For example, would it be the number of reads or how good the genomic coverage is for the suspected pathogen?
Yeah, I thank you for this interesting question. I think indeed you can approach this from different ways. One of the things is indeed looking at how much of the genome is covered in contrast to the number of reads is important. The number of the coverage is important indeed. The other things obviously is, I agree that you should do this for every different disease type. You should use your machine learning models and just collect data and see what's going on and make sense out of the enormous amount of data. The other thing that you can do is, because I showed you in the paper by others, and we've seen that too, that you can distinguish active microorganisms based on their RNA expressions versus the ones that are not active. So that's another way of looking at it. But of course, it's still challenging. Yeah, I agree with that. But there are ways to overcome this.
Okay. I'm sorry, that is all we have time for. We've reached the end of the session, but many thanks for joining us live and answering the questions and for giving a fantastic talk. Thank you.
I was happy to do it.
Great.